So in the previous part of this lecture, we talked about how meiosis is a special type of cell division that takes one cell and makes it into 4 haploid gametes. And so the main goals of meiosis are to reduce the number of chromosomes in half, as well as to create genetic diversity. And in this part of the lecture, I'm going to go through the phases of mitosis, or the stages, and then talk about how meiosis achieves these goals during this process. Right, and so first I really want to get you thinking a little bit about why it's important to produce haploid gametes, which can come together to form a zygote. Right, so meiosis is there to produce these sex cells that have one copy of each chromosome. What would happen to the zygote if gametes were not haploid, but instead diploid? Take a second, think about it, and come up with your answer. Well, what would happen is there would be too much DNA, right? So if a gamete was diploid, that would mean the egg cell has 2 copies of the chromosomes, and the sperm cell has 2 copies of the chromosomes. And so when these 2 gametes come together, they generate a zygote that has 4 copies of the DNA. And having too much or too little DNA is a bad thing, right? So it's sort of a Goldilocks thing. 2 copies is perfect. Too much DNA, more than 2 copies, can lead to disease or ultimately death of the zygote, and too little DNA can lead to the same thing, right? And so if you want to create a zygote that is diploid, you first have to generate haploid gametes that only have 1 copy of the DNA, so that when they come together, they generate a zygote which is has 2 copies, right? And so meiosis as a process has to have the number of chromosomes. And there are 2 stages of meiosis. There's meiosis I and meiosis II. And prior to either of these stages beginning, right, so prior to meiosis I, the DNA is replicated in S phase, just like it is before mitosis, because you are going to eventually divide into multiple new cells. So you need to replicate the DNA first, and then cells can enter into the first stage, which is meiosis I. Right? And meiosis I is organized into 5 phases: prophase I, metaphase I, anaphase I, telophase I. Right? And this should sound familiar to you because they're the same names of the stages in mitosis. Right? And it's the same order as well, with prophase being first and telophase being last. And I'm going to go into sort of the more molecular detail of what happens in each of these phases in the next couple slides. Right. And so at the beginning of prophase I, you have your DNA that's been replicated during S phase. Duplicated chromosomes exist. And what they start to do at the beginning of prophase I of meiosis is condense. You have a lot of DNA, and you need to condense it in order to be efficiently separating it later on. And so this loose, stringy DNA condenses into X-shaped chromosomes, duplicated chromosomes that look like X's, right? And then in prophase I, the homologous chromosomes, or the ones from each pair, the one from mom and the one from dad, find each other, and they pair up together and form these structures called synapses between them. And so in the process, those duplicated X-shaped homologous chromosomes find their pair and become homologous pairs, right? And so there's 2 X's. Those 2 X's contain 4 sister chromatids, or 4 individual chromatids that form what's called a tetrad. And then once the homologous pairs are aligned with each other in this tetrad, you can see 1 duplicated chromosome here in blue, 1 duplicated chromosome here in red. These 2 together would be a homologous pair. And those homologous pairs can undergo a particularly important event known as crossing over, where sister chromatids, or 1/2 of these chromosomes, can actually break and be swapped between the 2 homologous chromosomes. And sometimes this is also called recombination, right? And you can see that it's usually called crossing over because there's a physical cross made between these two arms of the homologous pairs, right? And so now at the end of crossing over, you have a duplicated chromosome that has chromosomes that look like the original, a chromosome that has a little bit of this red attached to it, one with a little bit of blue, and one that looks like the original. And what crossing over really does is it introduces some genetic variation. And then after crossing over, at the end of prophase 1, the chromosomes have to be free to move around the cell. So you need to break down the nucleus that contains them. A spindle needs to form, and the spindle, remember, is that structure that moves chromosomes around. And the chromosomes need to attach to that spindle so that they can be moved to the correct location, right? And so all of this happens in the first phase of meiosis I, prophase 1. A lot of stuff is going on. This is a very essential phase, right? Because this is when crossing over happens, and genetic variation comes from crossing over. And so then the mitotic spindle pushes and pulls the homologous pairs that were grouped together in prophase 1. You can see those pairs here, 2 greens, 2 purples. And in metaphase 1, those homologous pairs line up at the middle of the cell, or the metaphase plate. And then in anaphase 1, which is the next stage, the homologous pairs are separated from each other. One purple chromosome moves to the right, one purple chromosome moves to the left, same with the green, and those pairs are no longer together. And they get pulled towards the poles of what's going to ultimately become 2 new cells. And this is a process known as chromosome disjunction. The disjunction is a separation, and the chromosome is telling you what gets separated. In this case, homologous chromosomes get separated from each other, and the homologous pairs are pulled apart. And then finally, in telophase 1, everything that happens in prophase 1 is reversed. The chromosomes decondense, the nuclear envelope reforms, and the mitotic spindle dissolves. Because you no longer need the spindle, you've moved the chromosomes to where they're going, and you can reform nuclei around them and let them decondense before the next phase, which is meiosis II. Okay, and so at the end of meiosis I, the cells are officially haploid. They officially have half of the number of chromosomes that they started with, because you took one of the duplicated chromosomes from the homologous pair and put it into one cell, and one of the homologous pair and put it into the other. And since there's no additional kind of interphase between meiosis I and meiosis II, the DNA doesn't replicate again before the next set of divisions occur. Right? And so you have haploid cells at the end of meiosis I. They have one copy of the DNA, either mostly from mom or mostly from dad, and now they are haploid. And then they can enter into a series of 5 phases again. Prophase 2, metaphase 2, anaphase 2, and then telophase 2, and ultimately cytokinesis. And once again, same order, same names as meiosis I, except there's a 2 after all of them. And it's basically the same process as well. There are some major differences, and I'll highlight them as we go. In prophase 2, the chromosomes have to condense again. They have to form a spindle. There has to be a spindle formed. The nucleus has to dissolve. The chromosomes have to attach to that spindle. But what's important to note is that in prophase 1, there's no crossing over. So that only in prophase 2, that only happens in prophase 1. So you've already done crossing over in prophase 1, you're not going to cross over again in prophase 2. And one reason for that is because there's really no point, because both of these chromosomes are purple. And so if you cross this piece over with this piece, you would end up with the same exact result. So there's no point in crossing over in prophase 2. And then once the chromosomes are attached to the spindle, condensed, in metaphase 2, which is the next phase, they can be pushed and pulled and ultimately lined up at the center of the cell again, or that metaphase plate. You can see here, one duplicated chromosome in purple, another in green, perfectly lined up at the metaphase plate. And then what happens is a chromatid disjunction, or a separation of the chromatids from each other. And this happens in anaphase 2 of meiosis. And what you'll notice is that now one half of those X's, both purple and green, or one chromatid, is pulled to the left and the other chromatid is pulled to the right. That X-shaped structure is pulled apart. And the sister chromatids making up both sides of the X are separated and pulled towards opposite poles. And so at the end of all of this, in telophase 2, you now have sister chromatids in 4 individual nuclei that form around those chromosomes. The mitotic spindle dissolves, and you end up with 4 separate nuclei. But you'll notice that the cells still technically physically connected to each other. And so there has to be a cytokinesis step, or a physical division, because we've already divided the nuclei at the end, divided the chromosomes and the DNA at the end of telophase 2. But there has to be a division of cytoplasm, and that's what happens last. There's 4 nuclei, and then there's a physical separation of these 4 cells into 4 haploid daughters that are physically separated by new membranes. And so you start with 1 diploid cell, and through 2 rounds of division in meiosis I and meiosis II, you generate 4 haploid daughter cells. And at this end of meiosis II, the cells remain haploid, right? They still have 1 copy of each chromosome, and so that doesn't change. And to visualize this process, I have this GIF, right? And so this is the beginning of prophase 1. You can see crossing over happening there. The homologous pairs are going to line up at the metaphase plate in metaphase 1, and then they're going to be separated in anaphase 1 from each other. Telophase 1 is happening now. And then you enter meiosis 2. Those chromosomes are now metaphase 2, where the chromosomes are lined up, anaphase 2, where the sister chromatids are separated, and telophase 2. Right, and so we'll watch that again with a little more detail. So here is prophase 1 of meiosis. Homologs are crossing over. Those homologous pairs are lining up at the metaphase plate during metaphase 1. They're going to be separated from each other in anaphase 1, and then telophase 1. And then meiosis 2 can begin. Prophase 2, metaphase 2 where they're lined up, and anaphase 2 where they're separated, and telophase 2. And ultimately, cytokinesis to make 4 haploid daughter cells. And so the goal of reducing the chromosome number by half has been achieved just through the 2 divisions of meiosis 2, right? The DNA is replicated only once in interphase before meiosis 1, and because it's not replicated again in between meiosis 1 and meiosis 2, the chromosome number can be reduced from a diploid number or 2 copies in each cell to a haploid number to generate the haploid gametes that you need. And then the next portion of the lecture, I'm going to discuss how the genetic diversity goal is achieved during meiosis.